Hi, I'm Dr. Messina. Today I'm going to be discussing why is it that if they have fast-tracked the development of a vaccine for the SARS-CoV-2 virus, that we're going to have to wait a year to 18 months to see it being used in the general population. And the reason for that is because there's a lot of governmental oversight in the development and production of vaccines. Yes, we want it expedited, but we also want it to be safe and effective. I'm going to discuss the development of vaccines from the inception to the point where we are delivering it to the general public. And there are 6 stages of vaccine development. The first is the exploratory phase. The exploratory phase is done completely in the laboratory. You have cell cultures, and you expose those cell cultures to a whole variety of antigens in the hope that one or more of those antigens will create an immune response. The antigens could be anything from live virus to killed virus to proteins to bacterial toxins. And this part of the study could go on for 2 to 4 years. Once we get to the point where we do have an antigen causing an immune response in the cellular tissue, we could go on to the next phase, the preclinical phase. In preclinical studies, we are exposing these antigens to not only the cell cultures once again, but to the animal model. And we're hoping that exposing it to the animal model will cause an immune response, and further hoping that that immune response will then be provoked in a human. The leap of faith is that will the animal model and the human model respond in the same fashion? It's in this preclinical phase where a lot of potential vaccines are scrapped and discarded. Either they didn't cause the same immune response that they caused in the exploratory phase, or they just proved to be too dangerous. Once you have something causing an immune response in the animal model, you might be able to expose that animal to the disease and see if you got any type of clinical immunity to this disease state. And again, the animal model could take several years of study. Aside from looking for an immune response, we're also attempting to find what type of a dose is going to be required to create that immune response, and how frequently would you have to give this dosage in order to maintain immune responses. Should things go well in the exploratory and the preclinical phase, then the researchers present to the FDA an investigational new drug application, an IND application. And they will tell them what they were hoping to find, what the antigens were that caused this immune response, what type of immune response did they get, and also the safety profile. The FDA has 30 days to give a response, and should they say, yes, you can proceed, then you go on to the clinical development phase, which has 3 subphases. In the clinical development stages, we are now testing on human subjects. If this vaccination is intended for pediatric use, they still start in the adult population and they slowly work backwards until they get to the pediatric age group. So we don't start testing in children. There are 3 phases in the clinical development stage. In phase 1 of a clinical trial, we're dealing with a small patient population, usually 20 to 80 people, and we're looking specifically not only for safety, but to see if in the human studies we are still getting some kind of an immune response. Sometimes it doesn't happen, it just doesn't correlate to the animal model. In phase 2 clinical trials, we are now dealing with several hundred participants. And they too are given the antigen, and again, checking for immune response. In phase 2, aside from the safety measures, we're looking to see what type of immune response we got. Immunology is very, very complex. It's a series of chemical reactions leading to cellular reactions. And I'm going to explain what they're looking for in very, very simplified terms. Basically, when we're looking at immunity, we're hoping that we get some antibody production from B cells. Antibodies will go and hopefully attack a virus or a bacteria, or sometimes they go and they line the mucosal membrane, not allowing that virus or bacteria to enter the cell. We're hoping to get macrophage stimulation. Macrophages come and gobble up invaders, be it bacterial, viral, even tattooing. We hope to get some T cells involved. T cell immunity could be of three types. T helper cells modulate the immune system. T killer cells, just like the name says, kill things. And then memory cells. Memory cells are important for getting long-term immunity. Some vaccines don't stimulate the memory cells, therefore have to be repeated quite frequently, while some, as we saw with the smallpox vaccine, should give a lifelong immunity. And that's part of the memory cell. The goal in phase 2 studies is to, again, find the safety, find dosages, the immunogenicity of a vaccine. In other words, how potent of an immune response are we going to get? The schedule of vaccinations that are going to be required, and also the route of administration. Is it going to be an intramuscular shot, or is it going to be an intranasal spray, or by giving some other method? If we have a successful outcome in the phase 2 studies, we go to phase 3. Phase 3 is a large test group, tens of thousands of subjects, and it could last several years. The reason it's a large test group is we want to find out exactly what type of side effects can we get from these vaccines. So if it's a side effect that might affect 1 every 10,000 people, well, then we're going to need a minimum of 60,000 test subjects to find out exactly how often it's causing this problem or how severe the problem is. That's why phase 3 takes a long time and requires extensive study with an extensive patient population. Now, should we go through all of these phases and everything is looking favorable, then a biologics licensure application is submitted to the FDA. And this is important to know the history of viruses as to why the FDA has decided that it has to license plants to create the vaccine, and then it follows them very closely to make sure that there's proper quality control. And this goes way back to around 1902. In the early 1900s, we had vaccinations for cholera, and we had vaccinations for some of the plagues, and it was very much the Wild West. There was no oversight onto where these vaccines were created, how they were created, what was in them. People were getting overdosed, people were getting underdosed. Some instances had the wrong vaccine given to them because it was in the wrong vial. And the government got involved and started a biological agency to oversee this, which grew into the NIH. So we have a lot of governmental oversight and specific licensure for a laboratory or a pharmaceutical company to create and distribute these vaccines. There's one case in particular that's the reason for this quality control. In 1954, there was the Cutter incident, and the Cutter incident is the reason why the FDA is so involved in quality control. Cutter was a small family-owned pharmaceutical production company, and they started creating vaccines for polio, except they were dealing with live polio virus that had to be killed during one of the steps in the process of creating this polio vaccine. And the method they used did not kill it, and they exposed approximately 200,000 children to polio through the vaccine. And after that, the government got extremely involved in quality control. That's why even after they get the licensure, there's still going to be ongoing inspection of the facility and the steps used to create the vaccine and the potency of the vaccine. It has to be uniformly potent dose to dose. So it's a safety measure. So all of these measures that make the vaccine so safe and effective for us take time. And that's the reason why we're not going to see a vaccine, even in dire conditions, for about a year to a year and a half. I hope this answered a lot of your questions. And if you enjoy seeing these videos, please remember to click subscribe as well as the little bell to be notified when the next video is available. Take care. Have a great day.